Hello, welcome back to systemic embryology class sessions. So today's topic is on development of heart. We shall divide the development of heart as part 1 and part 2, where in part 1 we will just understand how the heart tube has formed and what are the various parts of heart tube. And in the second part we are going to look upon the interatrial septum, interventricular septal formations and formation of different chambers of the heart. So let's begin with today's session in development of heart part 1. So here in this image we can appreciate uh, a disc primitive with a primitive streak and septum transversum, and this is the oropharyngeal membrane, and here is the cardiogenic area. So the vascular system, if we see its development, it starts its development in the middle of the third week of gestation. So just during the formation of trilaminar germ disc, at the same time we see the formation of vascular system as well, and it is the first system to start functioning in the embryo. So the progenitor heart cells lies initially in the epiblast cells. So the blue colored structure here are the epiblast cells, which are the progenitor cells of heart cardiogenic area. So these progenitor heart cells which lie in the epiblast immediately cranial to the primitive streak. So the one which are near to the primitive streak. So this is the cranial end of the primitive streak. So just at the cranial end of the primitive streak, the epiblast cells which migrate from the epiblast layer through the primitive streak into the splanchnic layer of lateral plate mesoderm. So you might have known what is lateral plate mesoderm by now in general embryology. So these epiblast cells which are at the cranial end of the primitive streak, they migrate into the splanchnic layer of lateral plate mesoderm, which later turns to form a primordial heart. So that is about the basic idea, and thus the heart is, if you see its origin, it is mesodermal in origin, and the cells of the epiblast migrate through the primitive streak to reach the primordial cardiogenic area. So here is the primordial cardiogenic area. So here is the primitive streak, this is the primitive node, and here is the developing notochord which is in the midline. This is the oropharyngeal membrane or buccopharyngeal membrane, and caudally we can see the cloacal membrane. Both are bilaminar membranes, and notochord is limited anteriorly by oropharyngeal membrane. And here this area is the cardiogenic area, and this yellow color one what is developing here is the septum transversum. So the progenitor cells which lie in the epiblast immediately adjacent to the cranial end of the primitive streak, they migrate into splanchnic layer of lateral plate mesoderm. So that is the reason the heart is mesodermal in origin. So the cells of the epiblast migrate through the primitive streak to reach primordial cardiogenic area that contains cluster of cells approximately around on 16th day of gestation. Once the cells are established in the cardiogenic area, the underlying pharyngeal endoderm induces these cells to differentiate as myoblast and blood islands. So the underlying pharyngeal endoderm, which is near the cardiogenic area, that is this area. So here the endoderm, they will differentiate to form myoblast cells which forms the part endothelium, and also the blood islands that will form the blood vessels and blood. So myoblast will form the blood vessels, and blood is from the blood islands. And the heart develops from a tube. It itself is a blood vessel by development. So it is a primitive heart tube which is from the mesenchyme in the cardiogenic area of embryo. So this heart tube is formed within this cardiogenic area of the embryo. In the beginning, the cardiogenic area, it is located anterior to the neural tube. So this is the cranial end of the embryo. So it is located at the cranial end of the neural tube. So neural tube, we know it is just lies superior to notochord. This pink color line is the notochord. So just above the notochord is the neural tube. So cardiogenic area lies cranial end of the neural tube, and that is cranial to the oropharyngeal membrane, and it is a trilaminar germ disc between the septum transversum and procaudal plate. So it forms a cardiogenic area. It is an area in the cranial end of the trilaminar germ disc embryo. So we know by the third week trilaminar germ disc forms. So between the septum transversum, this yellow one is the septum transversum, and here is the procaudal plate or oropharyngeal membrane. So the part between them forms the cardiogenic area. So the part of intraembryonic coelom overlying in this area, it forms the pericardial cavity, and the splanchnopluric mesoderm underneath the pericardial cavity forms the heart tube. So with the development of the brain vesicles, that is the neural tube later on develops into forebrain, midbrain, hindbrain, and with the development of head fold of the embryo, we know the head will start folding ventrally. So because of the head fold of the embryo, the oropharyngeal membrane moves forward, and the heart tubes and the pericardial cavity lie ventral to the cervical region. So initially here in this image we can appreciate in the cranial end of the embryo. Later we will see in the next image how it shifts into the ventral side because of the head fold of embryo. Now with this image we will understand how the endothelial heart tubes and primitive heart tube has formed. So here we can see two endothelial heart tubes. Initially these endothelial heart tubes, they develop as a pair of longitudinal blood vessels which are called as dorsal aortae. So they are called as dorsal aortae. So the blood islands form a pair of longitudinal blood vessels called as dorsal aortae or endothelial heart tubes. So which are the earliest sign? So the earliest sign of the appearance is by two endothelial strands which are known as angioblast cords. These are called as angioblast cords, and which appear in the cardiogenic mesoderm, and these cords later they get canalized to form the endothelial heart tubes. These cords later eventually they get canalized to form heart tubes. So during embryonic folding, those endothelial heart tubes, they approach each other. So because of lateral embryonic fold, especially the lateral folds of the embryo, because of the lateral folds, the two endothelial heart tubes, they approximate or approach each other, and they form a single heart tube. And this fusion begins on the cranial end. So you can see the upper part of the heart tube is fused. So here is the single heart tube. So it begins in the cranial end and extends caudally, except the caudal part which remains unfused. So we can see the caudal unfused part. So the caudal part is the venous end, the cranial part is the arterial end. So the cranial part is the arterial end, and the caudal part is the otherwise called as venous end of the heart tube. So thus the heart tube becomes a continuous expanded tube with inner endothelial lining and outer myocardial layer. So the heart begins to beat by 22nd to 23rd days. So 22nd to 23rd days of gestation, we can hear the heart sounds, and blood flow begins during 4th week. So the blood flow begins during 4th week. So initiation of development of heart tube is during 3rd week, and it starts functioning by 22nd to 23rd days, and the blood will flow through the heart tube on during 4th week. So here is the head fold of the embryo. So as we saw in the previous first image, the heart tube was the cardiogenic area was on the cranial side of the procaudal plate, that is the cranial end of the embryo, which is caudal to the septum transversum. So here we can see the head fold had formed here. So because of the head fold, the cardiogenic area moves to the ventral aspect. So the cephalic end of the early somite here, where we can see the endocardial heart tube. This is the endocardial heart tube, and its investing layer, which is called as pericardial cavity, which is derived from intraembryonic coelom. So this is the pericardial cavity, and the dorsal mesocardium is breaking down here. So this is the dorsal mesocardium. We can see it is breaking down to form the heart. So the myocardium differentiates to form a thick myocardial layer, and also secretes a layer of extracellular matrix, which is called as cardiac jelly. So this myocardial mantle, it forms a cardiac jelly, and this cardiac jelly, it is rich in hyaluronic acid. So the surrounding mesenchymal cells differentiate to form epicardium. So the outermost will form the epicardium. So therefore the heart consists of three layers: endocardium, which is derived from the endothelial lining of the heart tube; then myocardium, which is derived from the cardiac jelly, that is myocardial mantle of cardiac muscles will form the myocardium from the splanchnic mesoderm; and epicardium from the visceral pericardium, and which is derived from the mesothelial cells of the splanchnic mesoderm, and also involves in the development of coronary arteries. So this epicardium also involves in the development of coronary arteries, which are considered as largest vasa vasorum of heart, because heart itself is considered as a blood vessel. So coronary arteries are the blood vessels supplying a blood vessel. So that is the reason it is vasa vasorum of heart. And the mesoderm around the endocardium, forms the myocardium. Mesoderm around the endocardium forms the myocardium. The myocardial cells secrete an extracellular matrix, we know, which is called as cardiac jelly, which is rich in hyaluronic acid. And if you see this cardiac jelly, which forms the myocardial mantle here, it is like a gelatinous substance, which is secreted by the cardiac myocytes in the present between the endothelium and myocardium of embryonic heart, and which transforms into the connective tissue of the endocardium. So the extracellular matrix of the heart, historically it is termed as cardiac jelly. So from the beginning it is termed as cardiac jelly, acts as a site of deposition of inductive factors from the myocardial cells, which later modified the differentiation of specific endocardial cells. So it has been called as gelatino-reticulum. Cardiac jelly has been also called as gelatino-reticulum, which forms the myocardial mantle. So myoepicardial reticulum, and also it is the other name for gelatino-reticulum, and otherwise called myocardial basement membrane. You can also say it as myocardial basement membrane. And this jelly, cardiac jelly, it accumulates the endocardial cushions, which are the primitive precursors of cardiac valves, the valves present in the heart. So the mesoderm that migrates from the coelomic wall near the liver into the cardiogenic area forms the epicardium. So that is about the formation of three layers. So one basic thing to know, endocardium is from endocardial heart tube, which forms the endothelial lining. Myocardium is from myocardial mantle, which is secreted by the cardiac jelly, and it is rich in hyaluronic acid in the beginning. And epicardium is differentiated as the outermost layer of it, which is from the splanchnopluric mesoderm. And this is the pericardial cavity. Epicardium is nothing but the visceral layer of pericardium. So that was formation of heart tube, and we saw the cranial head fold and movement of the heart from the cranial to the ventral side. And then we shall now discuss about the parts of the heart tube. So heart tube, if you see, it is an elongated structure exhibiting dilatations. You can see each one, nice colors are there. So which are like alternative dilatations. So there are 4 dilatations mainly, 1, 2, 3, 4. 4 dilatations from cranial to caudal. They are like bulbous cordis. So this is the outflow and this is the inflow. Outflow, this is the outflow, and this is the venous end, that is the inflow. Upper end is the arterial end, so it is outflow. So from down if we start, the lower unfused part is called venous end, which is formed by sinus venosus. This blue colored structure is the sinus venosus. Then primitive atrium, which is formed by this green colored structure, which is the actual atrium. Then primitive ventricle, this orange large one, forms right and left ventricles later. And bulbous cordis. Bulbous cordis terminally forms truncus arteriosus. So these are the parts of the heart tube. So truncus arteriosus forms the arterial end and it is continuous with the aortic sac cranially, and aortic sac divides into aortic arch arteries. So the truncus arteriosus, this point is the aortic sac where you can see 2 arteries arising from it, which are called as aortic arch arteries. Later on, each arch has got its own artery. So there are 6 pharyngeal arches, 5th one disappears completely. So remaining are 5 pharyngeal arches. So 5 arteries, 5 aortic arch arteries goes to each arch. Next about the sinus venosus. Sinus venosus is the venous end containing 2 horns, and each horn receives umbilical vein, vitelline vein and common cardinal veins. Next, regarding the truncus arteriosus, how it arterial end and the venous end we are going to discuss. So here is the cranial end which forms the truncus arteriosus, which is the arterial end, and it is continuous with the aortic sac. So this is the truncus arteriosus, this is aortic sac, and cranially the aortic sac divides into 2 aortic arch arteries which invade the pharyngeal arches. So aortic arch arteries. Then the sinus venosus, which forms the venous end. So this is the caudal end of the heart tube, venous end, and it contains 2 horns, right and left horns, and each horn receives veins from common cardinal. This is the common cardinal vein. The middle one is the umbilical vein, and most medially vitelline veins. So these 2 are the vitelline veins. The outermost are the common cardinal veins. The middle ones on each side are right and left umbilical veins. So as the heart tube grows, it blends, forming a loop which is called as cardiac loop, which invaginates later into the pericardial cavity. So let's see each dilation what it forms, adult derivatives. So from cranial to caudal, truncus arteriosus. Truncus arteriosus forms the ascending aorta pulmonary trunk. Bulbous cordis forms the smooth part of right ventricle, which is called as conus arteriosus, and smooth part of left ventricle, which is called as aortic vestibule. So bulbous cordis will form the smooth upper part of right and left ventricles, which are called as outflow part. Then primitive ventricle forms the trabeculated rough part of ventricle, which contains papillary muscles like bridges, ridges and pillars. We say different types of papillary muscles are appreciated in the primitive ventricle. which forms the trabeculated part of both right and left ventricles. Then primitive atrium. Primitive atrium is also rough in texture, which contains the trabeculated part of right atrium, which is and left atrium as well, which is mainly appreciated in the auricles of right and left atria, and also the musculi pectinata of anterior part of right atrium is also from the primitive atrium. Lastly, the sinus venosus. Sinus venosus will get absorbed into the developing heart and forms the smooth part of right atrium. It forms the smooth part of right atrium, which is the posterior part of right atrium, called as sinus venarium, and it also takes part in the formation of coronary sinus, oblique vein of left atrium. So these are the derivatives of sinus venosus in adult heart. So let's see how this heart tube will loop inside the pericardial cavity and forms a adult heart. So as the heart tube grows, it bends, forming a cardiac loop, and which invaginates into the pericardial cavity. So here is the pericardial cavity. Initially this is the dorsal mesogastrium, and here it is the pericardial sinus, which develops later. So we can see the heart tube starts invaginating into the pericardial cavity. So the cardiac looping, if we see, it is rightward bending and rotation of the primary heart tube that occurs over several developmental stages. So which all together forms in contact with the venous and arterial poles. So this is the venous and arterial poles. So you can see the venous and arterial poles, they will come in closer with each other. So the venous is the blue one and arterial one is the pink one in this image. So initially the tube is like this. This is the anterior end, this is the posterior end. So slowly the heart tube will approximate like this. So they will each end they will approximate and forms a complete heart tube like this. So because of the looping of the heart tube, the caudal sinus or the venous end turns posteriorly. So the venous end goes posteriorly and it will join with the arterial end posteriorly, and it gets absorbed into the developing heart, forming the smooth posterior part of the atria, especially the right atrium. The posterior part of the left atrium is formed from the absorbed part of pulmonary veins. So this is the basic idea about the looping of heart. So the anterior rough part of the atria are derived from primitive atrium, and the rough inflowing of the ventricles also derived from the primitive ventricles. They are from primitive ventricles. Smooth outflow of the ventricles are derived from the conus and truncus arteriosus. Truncus arteriosus, we know it divides into the ascending aorta and pulmonary trunk, and forming a septum between these two vessels, which is called as aorticopulmonary septum, which we would discuss in part 2. And during the 4th week, these dilatations, they undergo right side looping, that is called dextral looping, and which is completed by 28 days. So by 28 days we see the looping gets completed fully. So the atria gradually assumes the posterior location and the ventricles move to the more anterior side. So because of the tube formation, the posterior location will be forms the atria and anterior location is formed by the ventricle. So initially heart tube is like this, they will get approximated with each other. So the right horn of sinus venosus gets absorbed into the posterior aspect of primitive atrium to form sinus venarium, that is the posterior smooth wall of right atrium, whereas the rough anterior wall will form the atrium, right anterior part develops from atrium proper, that is primitive atrium. And the sulcus terminalis, which you see in the right atrium, which is at the junction of anterior rough part and posterior smooth part, indicates internally by the crista terminalis. So this muscular ridge, this crista terminalis, is along the right atrial wall from the opening of superior vena cava till inferior vena cava, providing origin to pectinati muscles. And talking about the left horn of sinus venosus, forms the coronary sinus. Left horn of sinus venosus forms the coronary sinus, and which opens into the posterior wall of right atrium. Superior vena cava, inferior vena cava develop from cardinal veins and open into the right atrium. Pulmonary veins develop in the dorsal mesocardium and open into the posterior wall of left atrium. And coronary arteries and associated veins are considered to be vasa vasorum of heart. So this is about a basic idea on the whole about the development of heart. So the opening of sinus, we can see the approximation here. So this is the inferior vena cava. This is future superior vena cava. This is the right horn of sinus venosus. This is primitive atrium. The front one is the primitive ventricle. We can see the sinus venosus gets absorbed, this one, yellow one, gets absorbed to form posterior part of right atrium, and the left horn of sinus venosus which gets regressed to form coronary sinus. It forms the coronary sinus here. This is from the left horn of sinus venosus, and posterior part will form the posterior wall of the right atrium. We can see the opening of superior and inferior vena cava here, and pulmonary veins gets absorbed into the left atrium here. So the opening of the sinus venosus into the primitive atria is guarded by right and left venous valves, and left venous valve along the septum spurium gets fused with the interatrial septum. This also we will discuss again. So as of now you can just see the site of opening of the sinus which is guarded by venous valves. The opening between the primitive atrium and the sinus venosus, that is called as primitive which is guarded by septum spurium, this one, and it has guarded by two venous valves which is called as right and left venous valves, which gets the right venous valve will get greatly stretched and it becomes subdivided into three parts by two muscular bands, that is superior and inferior limbic bands. So that is about idea of right atrium. So fate of various dilatations of the heart already we discussed. Truncus arteriosus forms a partition with the partitioned later on to form ascending aorta and pulmonary trunk. Truncus arteriosus is here. Primitive ventricle is here, and bulbus cordis. This forms the rough part, this forms the smooth outflow part. So primitive ventricle which later gets partitioned to form right and left ventricles, and primitive ventricle forms the rough inflowing part of right and left ventricles. Bulbus cordis forms the smooth outflow part of right and left ventricles. Next about the primitive atrium, this green one is the primitive atrium which is partitioned by a rough part of atria and to right and left atria later, which forms interatrial septum, and the central part of the right horn. So this is one is the left horn, one is the right horn. So the right horn, it gets absorbed into the primitive atrium forming the smooth part of right atrium which I have shown in the previous image, and the left horn of the sinus venosus forms the part of coronary sinus that opens into the smooth part of right atrium. So these purple colored structures are the umbilical arteries which carry deoxygenated blood into the placenta. So this is the umbilical artery. So this is about, and poorly oxygenated blood or nutrient poor fetal blood is sent back into the placenta via right and left umbilical arteries. This point to be noted. So this is an outline about primitive fetal circulation. So here are the vitelline vessels. Okay, let's talk about the development of pericardial sinus also. So pericardium has three components we know, that is the serous pericardium which contains the visceral and parietal layers. So visceral pericardium is from the splanchnopleuric mesoderm, or it forms the epicardium, and parietal pericardium is derived from somatopleuric mesoderm. Fibrous pericardium is derived from an inner layer, that is pleuropericardial membrane, which forms a fibrous partition separating. It forms initially a membrane which separates the pleural cavity from pericardial cavity. Later it gets fibrous to form fibrous pericardium. So remember, fibrous pericardium is from pleuropericardial membrane, and this fibrous pericardium covering the pericardial sac encloses the heart, and position of the heart tube in relation with the pericardial cavity. Before formation of the head fold, the endothelial heart tube lies in the floor of the pericardial cavity, caudal to the septum transversum, which we saw in earlier image also. And after the formation of the head fold, the pericardial cavity and heart tube comes to lie ventral to the foregut and cranial to the septum transversum, and with the heart tube lying on the roof of the pericardial cavity. So it lies in the roof of the pericardial cavity. Initially the heart tube will be ventral to the pericardial cavity. Later it forms the roof of the pericardial cavity, and it gets folded into this pericardial cavity. You can see it bulges into it, and it gets looped, cardiac loop, forming a complete heart. So this is all about the development of pericardial sinus, that is the pericardial cavity, how it forms, and pericardium, development of pericardium. Let's see some congenital anomalies as well. Ectopia cordis. It is a rare condition in which the heart lies exposed on the surface of the thorax. It occurs due to the non-union of two sternal plates. So sternal plates are not joined or not fused in this baby, exposing the heart outside. So it occurs due to the non-union of two sternal plates of developing sternum, and death occurs in most of the cases in first few days of birth. Dextrocardia is a rare heart condition in which your heart points towards the right side of the chest. So this was ectopic cordis. Next, about the dextrocardia. Usually the heart should be towards the left side, but here we can see it is directed towards the right side. So it is directed towards the right side, which is called as dextrocardia. So this is a very rare congenital anomaly. This is a very rare congenital anomaly in which the heart points towards the right side of the chest instead of left side, and it occurs if the heart tube bends to the left instead of to the right. Instead of dextro looping, if the left side it is loops, then it results in this formation. So the heart is displaced to the right, and there will be the transposition as well. And dextrocardia may be associated with situs inversus, which occurs in the abdomen, where all major visceral organs are reversed or mirrored from their normal positions. So the normal arrangement of internal organs is known as situs solitus. Please note down, the normal position of the internal organs is called as situs solitus, and reverse of all this is called as situs inversus. So with this I am winding up this session, and we would continue with the development of heart chambers in the next session. Thank you.